go. Hello, Chem 106. And we got protein. That is Mr. Universe, the Terminator, Arnold Schwarzenegger, showing off his biceps. And I'm sure his diet is loaded with lots of this good protein. So as I've mentioned, I taught a kids class for 10 years. And we initially called it Mythbusters. Joey actually came up with the name. And so we did different myths. And this was the myth about protein. And if you needed animal products for your best protein. And I don't know if any of you are old enough to remember this ad. I think it was from the 80s, but this was for Wendy's. And it was these three lovely women asking, yelling into the phone. I don't know if any of you even know, but this phone used to be how I would explain what an alpha helix was, because it has the perfect helix curves of an alpha helix. Anyway, they shouted into the phone, "Where's the beef?" Behind it is legumes, and they are amazing. Hopefully you're eating your legumes. And most of you will be watching this about the time of the full moon this week, which is May 7th. And which means that's about when, hopefully, if you start at the very beginning of our term, your first healthy change, you should continue with it for the next month and the next 30 days, second half of the term, and you can add a second healthy change. And so I would encourage you, maybe you'll be inspired by what we're talking about this week. How about legumes every day? It's tough, you have to meal prep, but a lot of you have the time and they're delicious. My favorite are these down here, the red lentils. And yeah, I could live on them, definitely. All right. So the myth was, do you really need meat to get protein? And so this past week, we saw that there was in the news the potential that the meat industry was going to be shut down, and there's all this meat that's rotting because the meat processors aren't able to work. And people were like, oh no, where will we get our protein? Well, eat your vegetables. And these are considered, yes, these are from websites that are very into the vegan. You can't see me here, but putting quotes, unquote. And part of this is, anyway, they're really high in all those nutrients. Look at the beautiful colors they have. And you've probably all watched the movie by now. There's going to be another movie for extra credit. There's one of several, and this guy was the strongest man in the world. He may still be, and he is vegan. And the movie is, what is the movie? Game Changers. So I actually have not seen the movie because it was only on, I think, HBO, but I think HBO is free right now. And it follows world-class athletes who have given up meat because it gives them a competitive edge. They don't have the fatigue they would get from eating animal protein. Highly recommend it. Not eating animal protein, watching the movie. All right, eating your nuts. Your nuts will give you great biceps. And there's, yeah, from Toy Story. Looking all the protein there from our legumes, vegetables, nuts, grains. Yeah, lots of protein. So what are proteins made of? A quick review of our previous slideshow, and hopefully your homework set, study set 5. Well, proteins, of course, are made of amino acids. This will probably be the first question on your next exam. It's a give me question. It's the biggest point of these two. Remember, this is correctly shown. You are always going to show it ionized. I actually had to look long and hard to find you that chart that's in your study set 5 that does show it correctly ionized. And the R group is the variant part, and we're going to be talking about that a bit more today. So there's about 20-ish. I say ish because there's actually more than that. Any compound in our body that has both the carboxy and the amino group is an amino acid. So there's a lot of specialized ones in our body. And they form a long chain, covalently bonded together, making them a polymer. And this polymer, of course, would be our proteins. And so it is, they are, this is back in the day when we could be within 6 feet of each other. All right, and there they are, those beautiful 20 amino acids. And if you're having any trouble falling asleep, well, you can just, instead of counting sheep, biochemistry grad students would count their amino acids. This was the trick I used to use when I was in grad school and getting ready for my dissertation. I would try and go through my amino acids. I don't think I ever made it even to valine. Like I said, I don't have sleeping issues, so obviously I passed. All right, levels of protein structure. This is a wonderful old slide because of the level of the graphics, but I kept it because it does point out, and this is the main point of today's talk. So we're going to go through these 4 levels of structure called primary, secondary, tertiary, and quaternary. And I just realized, you'll see it in the homework. Usually I'm writing on the board, and there's an abbreviation where you put a 1 and then a degree after it, and that means primary. Those you had Chem 105 when we talked about different types of alcohols, you saw me write it like that. So you'll see me write that shorthand in the homework. So secondary, you'd write a 2 with the degree after. So the primary is called primary because it's primarily the primary most important. It is our it's our protein chain. It is the peptide bond. And what is going to be distinct about this level from the others, peptide bonds are covalent bonds. Again, it is your shoulder attached to your arm, your hip is attaching your leg. They don't come off. No matter how much we jiggle and wiggle, and you can all jiggle and wiggle, you don't get to see me today, sorry. You only get to hear my lovely voice. So you can jiggle and wiggle, and your arm and leg and feet and toes, none of them come off. Secondary, tertiary, and quaternary structure are all attractive forces. So fancy word is electrostatic attraction, and it just means positives and negatives attract. So primary structure is covalent bonds. You cannot jiggle it apart. The other levels determine this overall beautiful structure. So it's artwork to a biochemist. All right, so let's embark on our journey. So primary structure starts with this man who won the Nobel Prize, apparently the year before Linus Pauling did, because I think that slideshow, the previous one, showed him in 1954. The very first amino acid sequence, protein in which the amino acid sequence was determined, was for insulin. And insulin, because of its importance for diabetes, type 1 diabetics, and once they found the amino acid sequence, it also then became the first DNA sequence that was determined, also by Frederick Sanger, and it was quite a feat and amazing. Notice it is shown correctly ionized at the end. That's the only place those ions will exist because everything else is covalently linked. The other thing that's interesting about this picture, this is a really small protein. It's only 51 amino acids, and it is the primary structure is just the order the amino acids go. And if you had 51 amino acids, there would be over 10 to the 81st. That's a 1 with 81 zeros after it. Different ways you can put these 51 amino acids together, and only one of them makes insulin. And so the order the amino acids are is of primary importance. And that was something I did not talk about, and now I don't even remember if it was on your previous study set. But if you have a number of amino acids, so let's say you have only 3 amino acids, you take that number factorial, so 3 times 2 times 1. So you take however many amino acids you have. So in this case, 51 times 50 times 49 times 48. And you keep multiplying it by every number smaller than that. And that tells you how many possibilities there are for the order you can put these in. And that's called, it's an exclamation mark. It's a factorial sign. You don't need to know it, but usually I talk about it when we're live in action and I have a blackboard right there for me. All right, again, covalent linkages. These are just some other cool proteins. And so insulin is small. This was the other thing I was going to say. There are some people, I am not one of them, I don't really care about this, but they say there is a difference between the term protein and peptide. And I am aware of the difference, but I will use them somewhat interchangeably. Don't sweat it. They mean similar enough. So some people say it is the number of amino acids. So if you have more than 51, you're considered a protein. So these 4 at the bottom. Insulin is considered too small, so chemical messengers, which are hormones and neurotransmitters, and we'll be talking about them next week, are usually smaller. This is not 100% true, and so most people don't use that distinction anymore. There are a lot that are only 3 or 4 or 8 amino acids, and so those are not usually considered a protein because they're just small. They're called peptides. And so you can see we get here to hemoglobin. This is huge. Remember water, when you learned about water way back in Chem 104, it was 18. The molecular weight was 18 grams per mole. You added up the 2 hydrogens and the oxygen. This is hemoglobin. You have a hemoglobin in every red blood cell, and you have so many red blood cells because they bring the oxygen around your body, and there's 65,000 grams per mole, because they're made up of 574 amino acids. And that's small compared, well, it's big, but there's bigger ones. Myosin, which is in your muscles, it's one of the ones moving your muscles, I believe is the biggest, and that's because it's these long fibers. Yeah, just kind of cool stuff. You don't need to know anything about that slide. All right, secondary structure. There he is, Linus Pauling. So he won a Nobel Prize. He won 2. Only one other person won 2 Nobel Prizes, and that was Madame Curie, Maria Curie. And so Linus won the first one in chemistry, and part of his prize, it was to do with determining structures of molecules, and especially this. He elucidated this idea that we have this helical structure inside of proteins, and it was due to hydrogen bonding. So the story is he had a cold, and so he stayed in bed. He was smart. So this was pre-coronavirus days, and that was a coronavirus, and he knew to stay home and stay in bed, not to get other people sick, and so he could get better. And he was probably eating his oranges because he knew all about that, and he educated us about it. And he asked his wife, this is pre-computer days. This is pre-electric typewriter days. And so he's just laying in bed, and he asked his wife to bring him a piece of paper and a pencil, and he just wrote out a long chain of amino acids. He didn't put R groups in. And what he found was every third one, that he cut it then into this chain, like this ribbon, and he twisted it, and he noticed that every third amino acid, if he twisted it just right, the C double bonded O that was sticking off would be attracted to the NH, 3 amino acids back. And that is an attraction. So NOF, if you remember NOF, I think I wrote it on the board when I was Tinkerbell. NOF is nitrogen, oxygen, and fluorine, the greediest 3 on the periodic table. Whenever they have a hydrogen attached to them, they have a huge polarity. And so there is an attraction between that hydrogen attached to the nitrogen and another oxygen. What makes secondary structure important is an area. It's not the overall structure, but it is an area of the protein with regular repeating pattern. And we're going to see lots of pictures as we go through this. And it is from the hydrogen bonding of the backbone. Whoever coined the phrase hydrogen bonding did us all a misservice, because it is not a bond. It is an electrostatic attraction between an area of positivity, hydrogen attached to nitrogen, and an area of negativity. So it's shown with dots, and so you can overcome it with jiggling, and then it's attracted back. So it's like magnets. So remember when you were a kid and you played with those trains? It is this pull, and then you could pull it apart, and then they're attracted back together, just how they were. All right, so it's amazing. It's how our body folds things into these beautiful structures. This is what he had to work with. Could you imagine? And his imagination allowed him to see it. So 2 types, you do need to know these. They're called the alpha helix, which is what is shown here, and that is Linus Pauling's genius. And also the beta pleated sheet, which I think is going to be the picture in the lower right. And so here you go. The overall 3D looks like that, and these are alpha helixes, and then the teal, bluish green ribbon are the beta sheets. This picture is interesting. This is a prion, and so the one over here on the left is, we all have these in our brain. So the prion is normal, and it's the alpha helix. And then as soon as one becomes abnormal and switches to a beta sheet, everyone after that is always abnormal. And that's where you run into problems. I have another slide on prions in a moment. I have first a slide on Linus Pauling and the alpha helix, which was his first Nobel Prize. The second one was not long after, actually, and it was the Peace Prize because he was against nuclear testing, and our wonderful government rewarded him by taking away his passport. And so he was not on the DNA Nobel Prize, or else he would have traveled and been able to help them figure out their structure. This is, again, what they had to work with. So he was making his R groups, and this is his way of showing, I don't know what, the hydrogen bonds. This is his wife, who he gives so much credit to, always says she was the person behind the scenes who allowed him to do what he did. And this is him lecturing. Again, no computers, none of this fancy stuff and imaging. He did it all, and his students had to have the imagination and see the hydrogen bonds. Yeah, and he was from Oregon. And his Peace Prizes are down at Oregon State in the library. That's probably not open right now. All right, and another example of the alpha helix is this. So we see it in actin and myosin. So in our muscles, we do see these long strands, and that would make sense. Or in your hair or your feathers. Any of you have your feathers? Our hair is looking awesome these days since, yeah. All right. And that's me showing off my muscles. That's a real picture. And yeah, I'm just really tough, in case you haven't figured that out. But I'm also showing off my alpha helixes, not just in my muscles, but in my hairdo, because my hair is always very kinky and curly. And that is because I don't have the disulfide, or I have the disulfide linkages that allow the curls. All right. This is a picture of beta sheet. They look like corrugated cardboard. So that's what it's trying to show here. This is a nicer picture, but you can see here, dot, 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 dot. And these are the dot, 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 dot. And that is because it is an attraction. It is just an attraction. You can jiggle and wiggle it apart, and we're going to get to that. And best example of an alpha helix is your curls. So any of you have curls, you can bling them right now. So I'm blinging mine, which I usually do in person. And the best example of a beta sheet is silk. And as females, we often glamorize and think, oh, I want something that's made of silk. And when I went to China, I got some silk shirts. And I will never buy silk again. I mean, it's beautiful and it's soft, but it has no breathability. It doesn't stretch. But spider webs are silk. They are beta sheets. And Spider-Man's outfit, I don't actually know what it was made out of, but silk is a protein. It's lightweight, it's flexible, and oh, remember, you all have to have a topic. This was a topic a student had. The slide is summarizing it in one. So you will be doing a presentation on Zoom. You can use like Google Docs, because there's a way we can share the screen, and yeah, pick a topic that excites you. I had my students last year often pick a topic because they could do really fun pictures in their slideshow. I don't know. So it provides a high degree of protection, strand for strand, 3 times more flexible, and far less weight. And so it is the new Kevlar, 5 times stronger than steel. And it is, silk is different from different types of spiders. So it's capable of absorbing a huge amount of energy. So it's a specific type of silk they discovered. But these spiders that make the silk, they don't play nice. And when you have a couple of them around each other, they just spend all their time fighting instead of making silk, and it just doesn't work. And so what they've done is they spliced it into other animals, including the silkworm. And silkworms play nice. They like made it through kindergarten. They know the golden rule. The weird one, and I don't have a picture of it because it was really weird. You can Google it. You can stop me right now and Google it if you want. They spliced it into goats. And then they milk the silk out of the goat. It was very disturbing to me. Aren't you glad I shared that with you? So you can stop me and go and look if you want, but you've been warned. All right, and it's biodegradable, and we can use it, yeah. So if we make silk from spider webs, it would be amazing. The whole thing with the coral reefs, airbags, parachutes can be made or are made. I guess parachutes are nylon right now. Yeah, or artificial tendons. So amazing. Sutures would be made from this. Really cool. All right, so this was, those of you who know Kenna would be laughing right now, is Kenna picked to do prions last term. So everybody was doing their presentations on like ginger or something. It was very positive. And then Kenna got up there and she just said with her big eyes, oh, I'm going to be the downer presentation. And that is because she did prions. And prions are mad cow's disease, in case you don't know. And as I said already, this is a normal prion. We all have them in our brain. This has already won 3 Nobel Prizes, and it was only discovered less than 100 years ago from a group of people in this very remote island who ate dead when their relatives died. It was a thing of prestige to eat them, and the women ate last, and so they ate the brain, and then they would get sick. And it was because if they ate somebody else who had a misformed prion, their brain started misforming, and what you end up with, normal brain over here, and this is a brain with lots of holes in it, which is not good. It's going to mean your brain can't do what your brain needs to do. And so again, healthy, and this is the unhealthy brain. Yeah, and so you can get it from eating contaminated food. They estimate, there's a guy in New York, this is 10 years ago, and it's not until the person's dead that you can look at the brain to see if it was Alzheimer's or mad cow, but it turned out that 50% of people diagnosed with the Alzheimer's, actually it was that. And in young people, because your brain works different, it is a very fast, like within 2 years. In older people, you just slowly, we have more connections in old people like me, and because of that, we're able to go around the holes and stuff. But, or just don't eat meat, and then you don't have to worry about it. There's also a genetic, but you would know if you had it because your whole family would be crazy and die at 40 because they would lose the capacity to sleep. So when people tell me they don't sleep, that's not true. You do sleep, you're just not sleeping well, because if you don't sleep, you go completely crazy. So as a family in Italy has this, and they're apparently the most beautiful people on the planet, and so people keep wanting to marry them, even though they know that these people are going to go completely insane when they turn 40, just to be part of this family because they're so gorgeous. Isn't that fascinating how we as humans are? All right, let's move on. Tertiary structure. It's that overall beautiful, look at that. It looks like a hot mess, doesn't it? Kind of looks like my hair these days. Well, okay, my hair on any day, but this is actually beautiful. It's not a hot mess. This tertiary structure is the overall 3D look of the protein. Your hair is protein, so its overall 3D look is its tertiary structure. So we can see areas of the alpha helix, secondary structure, and then the whole long chain stuck together as covalent one long chain is primary structure. And what is holding the tertiary structure together, and this is important, is the R group. We're going to have attractions from one R group to the other. We're going to walk through those. We've already seen that. Tinkerbell walked through them for you, but we're going to see them again. So shape, the overall shape determines the function. Remember all those functions we went through? So this is not part of your muscle. Your muscle would be long, fibrous ones. This is the globular hot mess. So I don't know, this could be your immune system, which is not a hot mess. Your immune system is beautiful. Here's another one. And right, determines if you're fibrous or globular, and our proteins fold. And so they're made as that long strand, and then they fold. This is part of the magic. They're still trying to figure out what determines the folding. There's different ideas, but it's an electromagnetic signal, my humble opinion. More than an opinion. But, all right, there's another one. So alpha helices, beta sheets inside, and then the overall is the 3D. So tertiary interactions. That's how you write tertiary. So you write a 3 with a degree. All right, this is the overall 3D, and you have all these different interactions. So let's walk through them. First type is a disulfide bond. So shown down here is a sulfur to a sulfur. So these were originally an SH. This is one cysteine to another cysteine. So these were SHs, and oxidation means losing electrons. So remember Leo. It also means you lose the hydrogen. And so those SHs lost their hydrogen, and so they bond to each other. That is a covalent bond. It is a line. That is very important. So the disulfide bridges up here, there are lots of them in your brain. Lots and lots in your neurotransmitter receptors. So the receptors for all your neurotransmitters have lots of these. Remember that when we get to the next slides. All right, second type is a salt bridge, and this would be an acidic side group to a basic one. So because it's ionized in our body, the acidic ones have a negative, and the basic ones have a positive. And so you get an attraction. It's ionic. You will see it called salt bridge. You may see it called ionic bridge. I learned it as salt bridge. And so that's the picture up here, a positive to a negative. Remember, all the positives and negatives in the backbone, that's what the squigglies are, they're gone because they made a covalent bond, the peptide bond. All right, the third type here, C, is an H bond. So if you have a polar side group to another polar side group, you could also have an acidic side group, because this guy's got this double bonded oxygen also to a polar one, or you could have a basic one to a polar one, because it says an NH. So just depending on how things fold and which ends up near another one. So on your table that was in your study set, it should have said, called them non-polar, polar, and then like polar acidic and polar basic. So the acidic and basic ones are still very polar, but we take them one step further because they become a true positive and negative. All right, the last one is not actually an attraction. So they don't show any dots. Non-polar things are not really attracted to each other. It's just that they don't like any of the rest of these, so they're staying away from them. So it's like, I'm just gonna stay in my own place, my own little cave here. It's a hydrophobic cave, or the hydrophobic closet, the hydrophobic pocket is, I think, what some of the books call it. So there's our hydrophobic. They hide in the center. They don't want to be out here around the edges because that's where the water is. So they fold into the center and they make that beautiful 3D. They're extremely important. Because of their fear of water, they're not wanting to be around water. They all end up in the center of our protein. All right, those are just pictures of overall. You can see you get totally different shapes. This one kind of looked like a monkey wrench to me. I don't know why. You get these beautiful stars if they look down on them. This would be a receptor one, so this would be crossing the cell membrane. So these in the middle would be all your hydrophobic. And you can see in all of these, there's usually areas of secondary structure. And it's the overall that gives you the tertiary. All right, this would be the insulin receptor. Insulin would just be like this little thing up here. Insulin's so small compared to its receptor. All right, And then the last one is quaternary structure. This does not always happen. It is when you have exactly what the slide says, 2 or more polypeptide chains that are interacting. Now notice it does not say covalently bonded. They're interacting. They're attracted to each other. This is hemoglobin. Hemoglobin has 4 units, 2 alpha and 2 beta. And the overall thing, you have one of these in your red blood cells. The center is called a heme group, and that's the part that's attracted to the oxygen. And so it's an example of quaternary. Not everything has quaternary. Another example of quaternary structure was collagen. I don't know if you remember from the previous slideshow, it made the triple helix. So it was 3 strands that are braided together and making a triple helix. So that was an example of quaternary structure because it was 3 strands. All right. So review, primary structure is the peptide bond, the covalent linkage, the order of the amino acids. This is the C terminus to the N terminus. All right. Alpha helix and beta pleated sheet are examples of secondary structure, just an area of regular pattern. Tertiary structure is the overall folding. And quaternary does not always happen, but it can, and it's when you have multiple subunits. Secondary, tertiary, quaternary is attractive forces. The one exception was the disulfide that we'll look at. This we're going to see more in the next slideshow, which is going to be about enzymes. That a simple protein is just amino acids. A conjugated protein has an extra group. So in our previous, there we go, it folds up. This was hemoglobin, so this is that extra group. And so this is a conjugated protein. All right, let's go to there. And so it's just got something extra. And we'll see that when we get to enzymes. All right, so we're gonna look at 2 words now. The first one is hydrolysis. This is an enzyme that eats proteins. And so it doesn't actually eat it, it breaks it into single amino acids. So it's cutting it apart. So hydrolysis means water cutting. So we saw that with glycogen back to the first week when we did sugars. So glycogen is long chains of glucose that our body stores, and then it gets cut down into single glucose. And so that is what an enzyme does. It completely breaks it from the protein. So we're cutting it into pieces. Now, a note for you is proteases don't actually start at the end and cut one at a time. There are only a couple that we have, and they can only cut between certain amino acids. And so Mother Nature put everything together in a way that the proteases would be able to cut them down. And then we've gone in and think we can do it better, but we don't have the enzymes that can cut certain amino acids. So you can't cut, say, between valine and alanine. But that didn't matter because Mother Nature never put the 2 of them next to each other. And then we did with our hybrids and stuff like that. All right, and so an example of hydrolysis, the problem is that proteins are tertiary structure. So it's like a ball of yarn. And so this little kitten's playing with a ball of yarn, and it's not going to be able to cut it with its claws. Really not efficient, right? And so this guy, he's still going for it. He's trying, but just it's really not working well, is it? So for hydrolysis to work well, we first need to denature the protein. And you've all seen this word. And so here we go. This little kitty's going to pull it apart. You can enjoy the video. So this is our normal protein with tertiary structure. Nobody's listening to me. You're all watching the cat. I know it. It's great. That's why I put the video in there, because you're all watching the cat. I could do something really weird right now and none of you would catch it. And denaturing means it unwound it. But there is an important but here. It still has primary structure. So the string is still all attached to each other. Our kitty cat is only denaturing it, not doing it the best way. We have better ways to denature. And then the protease would come in and clip it. So we have to unwind it so that you can clip it. All right, so how can you denature proteins? This, my dear students, is your lab. Well, I don't know what week we're in. I think we're in week 5. So this is week 5 lab, is you're going to denature protein. So milk and eggs. So the eggs, you're going to do it 6 different ways. One way you're going to have to improvise, and you just write down what you tried. Because in lab we use silver nitrate, and most of you probably don't have bottles of 1 molar silver nitrate hanging out in your house. So you got to try something. But you want just the egg white, so you're going to separate the egg. You can give the yolk to your dog, or it makes their fur extra bright and shiny. And then the white, you're going to split into 5 or 6 different containers, whatever the lab tells you, and then you're going to do this. So this you do need to know. There are 6 different ways in this slide that I could think of that you can denature a protein. You need to know what the 6 ways are. Again, you have your notes, so take good notes on this page, and how they work. So denaturing, again, we are not cutting, we're just wiggling it apart. So denaturing is wiggling and jiggling. So think of that cute little kitten on the last page. So the first way is simply agitation. So you're agitating, you're jiggling and wiggling. So if you make meringue, meringue is not actually really healthy because it's just loaded with sugar. Second way is heavy metals. So if you play heavy metal music like Iron Maiden, you'll probably jiggle and wiggle it apart because it's so loud. But this is mercury, or silver, or lead. Those 3 are the big 3: mercury, silver, and lead. And they denature because they love sulfur. Mercury loves sulfur. And so, where did we see sulfur on those previous slides? The disulfide bridge. And the disulfide bridge was special because it was actually a covalent link. But if you're exposed to mercury, silver, or lead, you lose the bridge, and that covalent link is no longer there. And does anybody remember where I said you have like zillions and zillions of these disulfide bridges? Oh, the receptors for your neurotransmitters. What makes your brain work and so beautiful and stuff. And so Johnny Depp is shown here because he beautifully portrayed what happens when you get heavy metal poisoning: mercury, silver, lead. There's other ones that can do it too, and they test for it. And so they were called Mad Hatters. So hatting back 150 years ago, when they made hats, they were made out of felt. And to make the felt just the perfect texture, they used mercury. And so the hatters all became crazy because the mercury ended up in them and in their brain, and it bound, and they all went crazy. You've all possibly heard of lead poisoning. So back when I was a kid, lead was used in gasoline, lead was used in paint. Lead is like amazing, these things it can do, and it's no longer used because people were going crazy. It's one of those things about the Roman Empire. Once they figured out how to make their aqueducts, which was this amazing feat, they were making it out of lead piping. And then they all went kind of crazy. You started having some pretty crazy emperors, and then you started having people being eaten by lions and other people watching it and cheering it on. I mean, that was not the beginning of the Roman Empire. They didn't like throw people into the lions and all watch. That's totally bizarre. Anyway, so mercury, lead, silver. Number 3 is alcohol. And so you're not going to drink it for the lab. You just add a little bit to your egg. I don't know that beer will work. I think you're going to have a higher percent. So rubbing alcohol or your hand sanitizer is alcohol, whatever alcohol you have in your house. And it denatures because alcohol, whether it's isopropyl alcohol or ethanol, is got OHs. And OHs love other OHs, which means hydrogen bonding. So it's going to make the hydrogen bonding ones, the polar ones, hydrogen bond to it instead. And so it's going to unravel it. All right. All right, number 4 is acid. So it could be lemon. You're not going to pee on your egg. So please don't do that. And please, if you do, don't tell me you did it. Just don't do it. You can use lemon, you can use vinegar, you can use anything that is acidic. I actually have another slide that gives you other ideas. If we can get lemons again. It's not like the first 2 weeks where lemons suddenly became disappeared. All right. So pH, you change the pH. The acidic side groups were missing a hydrogen, right? They were COO negative. So one O was double bonded to the carbon, and one was just an O with a negative. When you drop the pH low, a low pH means there's extra hydrogens around. And those extra hydrogens will fill in wherever there was a negative. And so you lose the negative. And so you can't have a salt bridge. So changing the pH disrupts the salt bridges. Agitation and pH is what your tummy does. So when you eat, your stomach is a pH of 2. Really, really, really low. In the lab, you only have to get the pH down to like 4.5. So 1,500-fold difference. And then your stomach gurgles, right? It moves the food around, and so it's denaturing the protein. So the protein gets gurgling, so it's agitating, and then we're breaking all the salt bridges. All right, and so in our stomach, we denature it, and the proteases are released, and they will then hydrolyze it. All right, way number 5, the 5th way to denature is detergents. I recommend detergents over soap, but I would like you to write down what you tried and how well it worked, because, and you just need a little bit. In lab, we have really cheap soap, because community college, and it doesn't work so well. And so when we do get to do this lab with real in live together, again with my students, I would like to be able to give them a good suggestion of which ones work. So you can use soap, hand soap, and you just put a little bit in there, and I had a whole lab on soap, week 3 lab, and its soap has hydrophilic and hydrophobic. Hydrophobic! Yeah, remember the hydrophobic pocket? Those are hiding. And now they go, oh, it's safe to come out. There's somebody out there just like us. And they come out from hiding. And the whole thing unravels. It should be really good at denaturing proteins. All right, and then the 6th way is you cook the egg. You can boil it. You can fry it. You can put it in your microwave. That might work. Somebody try that. Now you're all going to try it. You can poach it. It doesn't matter. You just got to heat it. And what does heat do? Heat makes it jiggle and wiggle, and it jiggles and wiggles, and the attractive forces and the secondary structure all comes apart, because secondary structure is all hydrogen bonds, and parts of the tertiary structure come apart, and you don't have to break every single thing. But the thing that doesn't happen with any of these jiggles and wiggles or hydrophobic or pH change is you don't break the peptide bonds. So the primary structure is still there, the ribbon or the yarn is still one long thing, and then you get the hydrolysis, cuts that apart. All right, so the other part of the lab is you are going to make cheese. So you're going to curdle milk. It does have to be cow's milk. My other class is doing this weekend, and there we go. I have my cow here. He's like, yay! And you're going to curdle the milk. And this is, I had to go through my slideshows. I think I have one that has a kid in it, but I've had to cut out so many pictures I have. But this is from, I've done this with the kids class. So we made cheese. I think this is supposed to be a rat. I'm not this good a drawer. There's a cat. That's me doing something with the kids. And so what's in your milk? Well, it's mostly water. And if you get low-fat milk, it's just even more water. It just dilutes out the fat. But it does still have, these are the proteins, number 4 and 5. 6 is a protein, and right now I don't remember what that stands for. And if you care, you can Google it, doesn't really matter. But these are the proteins in milk. So milk has carbohydrates, proteins, and fat. So for a baby, that's why mother's milk is perfect. All right, so what are the proteins in your milk? Well, do you remember Little Miss Muffet sat on her tuffet? That's her tuffet, eating her curds and whey. And along came a spider. Oh, and the spider said, what are curds? Or maybe Little Miss Muffet said, what are curds? Well, look at that. That's a protein. Look at the alpha helix. Isn't it beautiful? You could just do artwork of proteins. You should make that part of your lab. That anybody who's an artist could make some beautiful artwork. Tillamook Cheese Factory was the #1 visited place in Oregon, and they just made it even bigger. And that's probably not, I don't know if that's our cheese factory, but curds are casein. Is the chemical name, I guess, for the protein. And a lot of bodybuilders, I don't know why you would want your body to look like that, but they're obviously doing more than just casein. They're doing things that are going to make them not live very long. That's a picture of Joey 10 years ago, and this is long lost distant relative probably. This is high up in the Himalayas, and they all call Joey their little brother. This is the cheese factory. Very different look than the Tillamook Cheese Factory. Huge copper pot. This place was actually destroyed in the earthquake 4 years ago, but I'm sure they rebuilt it. There were so many people, and you had to hike for days to get to it. And then we stayed there for actually like a couple days in the tea house next door, because we then did a side trek. And Joey and I got to spend a day with them in the cheese factory, and they were so kind to us and made us lunch, and we couldn't say no. And yeah, I actually had to say no because I'm pretty sure the lunch was chicken and I was vegetarian. All right, casein is also the bristles in paintbrushes. Go figure, right? And the bristles in toothbrushes. I'm sure it's made from a plastic now, but the original toothbrushes, that's how they made them. So you might know where we're going with this. It is also whipped cream. And there is a reason Elmer's glue has a cow on it. Glue is casein. And I actually did this with the kids. And I might put it, if I remember before I post your lab, to put in the step to change your casein into glue, and then you can change the glue into flubber. I think I have a picture of me doing it. And there, cheese. Of course, you can make cheese. All right, cheese you're going to make is going to be the soft cheese. So most of you probably already knew about lactose intolerance. Maybe many of you already say, oh yes, I know I'm lactose intolerant. You are all casein intolerant. This is a bigger deal. There's a reason nobody knows it. So don't go to the dark side. Stay in the light. Casein is difficult to digest. And if you've been listening, it is difficult to digest because the order of the amino acids is not proper for us, our stomach. So it is animal, it's in the animal, not mother's milk. And so cow's milk, it's meant for the baby cow. And he has the enzyme that can cut it correctly, and we don't. And so you end up with tripeptides, and you get a lot of mucus. So it looks like mucus, and you end up with mucus. I remember my first week in acupuncture school, and the teachers all would talk about that. Oh, somebody comes in, oh, Americans, so much mucus, so much phlegm, so much dampness. Oh, just like they said, it was like walking into the ocean with your blue jeans on, and you just feel like that. You just feel like you're soggy all the time. And they said, oh, first cut out dairy, cut out all dairy. All right. Respiratory problems. And it's become even worse with the ultra homogenization. So homogenization is when they shake and jiggle the milk. So if you've ever seen milk that fresh comes out of the cow, it actually separates because the fat particles and the protein and water separate. And so homogenization made it all look smooth and look the same, but that breaks it up some, and that changes some of the structure and makes it even harder to digest. And yes, the spider was actually trying to protect Little Miss Muffet. And as I mentioned, partial digestion leads to leaky gut syndrome, gas, constipation, bloating, diarrhea, and a lot of skin disorders. There's other we already talked about. having a fatty acid, essential fatty acid deficiency, and also dairy. A lot of other issues, a lot of, for women, menstrual issues, pretty much you give up dairy and within the first cycle, they're all gone. I was skeptical hearing, but I've heard so many stories now, and I've actually mentioned this in class, and then I have students. One gal had rosacea, so she had other issues, and she made all these diet changes, but it was the rosacea, so it's where your face is red. And she gave up milk, and she said within a week it was all gone, and it never came back. It was her 30-day healthy challenge, and she said, I'm over dairy. I don't even miss it. The first week you miss cheese. Cheese is the one that is hardest. It was the last one that I gave up. It was the last one. I remember Joey's like, nope, not gonna give up cheese. He gave up everything else. My mom, but you give it up, and once you get over it, and the reason why it's the hardest one, it's the most addictive. It is actually a casomorphine. Yes, that is the word morphine. Extremely addictive. It binds your opiate receptors. So here's our cow. Everything in moderation. That's what she's saying. And let's just talk about the opiate receptor for a moment, in case you're not familiar with it. It deals with pain. It is our response to pain. It is how we perceive pain. Morphine, heroin. Look at the shapes. They're almost identical. They bind to it. And a perfect fit will give you the highest opioid effect. Oh, with no withdrawal pain. But all of these have withdrawal, serious withdrawal symptoms. So they must not be the perfect fit. Oh, and oxycodone, the ones that the doctor prescribes, highly addictive because you have no pain. Receptors for this are found all over our immune system. Think about that. Our immune system, which is needed for other reasons, it's binding to pain medication, and it probably changes how your immune system's working. It also is in your digestive system. Fascinating, because so many digestive issues, and right now so many immune issues, so many. Oh, so they found the receptor first, and they said, why do we have receptors? We must have a molecule for it. And then they found the molecule. So the beta endorphin's really cool. It makes a full circle. And this is an amino acid sequence. It's 8 amino acids, I think. Maybe it's 12. No, I can't remember. And I believe it is this part here when it's folded. It's this part that is what they're all mimicking. And so it fits into the receptor. But you know, even if this is folded, it doesn't look quite the same as all of those. And so you don't have withdrawal from your own endorphins that your body releases. Chocolate releases endorphins, by the way. Just had to plug that. All right, naloxone, in case you don't know, it actually blocks the receptor, a little bit different, you can see. And it's what they give to heroin addicts. And fascinating point, the one place in your body that does not have the receptor, I'm sorry, not this receptor, but has no pain perception is your brain. Your brain doesn't actually feel pain. Kind of cool. All right, this guy is amazing. I highly recommend you Google him, listen to his TED Talks, listen to his other talks. I have not had the chance to read this book. It was on my list, and then I've been a little busy preparing for your classes. So Dr. Neil Bernard, he is extremely charismatic and a phenomenal speaker, really good speaker. He is vegan, grew up on a meat and dairy farm in North Dakota. So he's a North Dakota boy, and I think that's why. He talks nice and slow, and he really gives good explanations. And he wrote this book about the cheese trap and talks about why it's so hard to give up cheese, because it's an opioid. So back here, it's really hard to give up morphine and heroin. Hopefully none of you are heroin addicts. Or if your doctor gives you oxycodone or any of the other ones, it's pretty amazing. And cheese, yeah, so you give up cheese. By the way, chocolate is also an opioid, and I will admit I'm addicted to that one, but I figure it's better than heroin, so I'm gonna stick with chocolate as my opioid. So what is the whey? It's another protein that is isolated from milk, and it's added to everything. So when they make the casein, when they make the cheese, casein is the protein that's in cheese. This is a byproduct. So we had to figure out something to do with it. So we sell it to guys who want to be more buff. And if you look at any processed food, it has whey protein added. It's not real because it's like been isolated. There's a picture of it. You see alpha helix, little beta sheet, overall 3D, held together by tertiary. So after the milk is curdled, you guys are going to throw the whey away. Okay. It's going to be strained out with the water, liquidy stuff. That's its name, lactalbumin. And the question, is it healthy? Well, studies show that it makes your muscles really big. And it's high in these amino acids, so they're branched, and increases muscle protein, weight loss, body fat, blah, blah, blah. It's one of these infomercials, right? And at the end they say, oh, wait, it might cause cancer. Excuse me? What? It might cause cancer? Is there a relationship between cancer and diet? Yeah. And so post-World War II, they looked at Japanese men and they were kind of floored. 25-fold, not 0.25-fold, 25-fold increase in prostate cancer after World War II. And they were saying, oh, it had to be Hiroshima, had to be blah, blah, blah. No, this was all over. And it had to do with they adopted our diet. We went there to rebuild and we said, oh my gosh, we have to eat meat. We have to eat eggs. Yeah, so the 7-fold increase in eggs, 9-fold increase in meat. Oh, and holy cow, 20-fold increase in dairy consumption in Japan post-World War, and almost a perfect increase in prostate cancer. So somebody somewhere, I didn't put a good reference here, dripped organic cow's milk onto prostate cells in a petri dish. They repeated it 14 times because they said, what do you mean? We keep seeing the cancer cells like want this stuff. The cancer cells are going, this is like almost as good as sugar. And they did it with almond milk and they actually found it decreased the cancer cell growth. So interesting. And then a meta-analysis is where they do an analysis of a whole bunch of like all the studies that have been done. It's a computer analysis. And they found, yep, there is a link between dairy and prostate cancer. And whenever you look at any of these books that talk about the top 25 anti-cancer foods, it looks like they're all plant-based. Or superfruits. Yeah, pomegranates. Season. So how did Little Miss Muffet's mom make curds? That's how you're going to be making them. So this is just a little background in case he informs my cells. Remember my cells? There were these boats. So this is a picture of what apparently a micelle looks like in 3D. And if we drop the pH to 4.5, remember the acid and basic side groups are no longer interested in each other, and the whole thing comes apart. There you go. It's unraveled. Wow. Isn't that amazing? Just that change in pH. These, so you can see there's still some attractions, but it changes it. And you get curds. And if you have a strainer, some of you might have to use a spoon. You're going to take a picture just like that, and you're going to have to be in your picture. So make sure it's a selfie. And then you can try it. So it denatures it. It jiggles it apart, right? And so you do first warm it up because we want it jiggling so it's ready to be denatured. And that's how we make cottage cheese and sour cream. They actually use an enzyme that they isolate from the stomach of baby cows to make the hard cheeses. So you can then feed it to your cat. You can try it on crackers, or you can make some dice with it. Take a picture and say, look what I did. Or you can make a Cookie Monster or a cow. Or you can make buttons. Yeah, before plastic, before the end of World War II, when plastic started coming out, so in the early 1900s, that's how they made plastic ornaments, buttons, decorative buckles, beads, jewelry, fountain pens, the backings of handheld mirrors, and the fancy, dancy comb and breast sets, and The Queen Mary of England. That's how they made her jewelry. She actually had plastic milk curd jewelry. I'm pretty sure some of these are not, but apparently some for jewelry. Yeah, that's the guy. When you used to go on the tour of Buckingham Palace, this little dude down in the basement, he's making her little plastic jewelry. And that's me working in her basement. This is me in the chemistry labs for, remember those days? And I did this with the kids. After we made the milk curds, I then was making, what's it called? Flubber slime. And the kids would not touch it. You have to touch slime to get it to work. And so Dominique was in there, she was making bird food across the way, and everybody was just looking at me like, what are you doing? But the Queen's very dignified. That's this guy. All right, so skim milk, again, we already talked about it. We removed the water, not the gnomes. And I have this slide here because we used to not be able to get the casein from the skim milk 4 years ago, and then suddenly last year it started working again. So I think that they were doing that ultra homogenization and something else, I forget what it was called, and people were really upset about it because they said, this is not good for us. And they suddenly removed it, but we couldn't get the casein out of it. And now we get this past year, every time I've done this lab, we get the same amount of casein, whether it's skim milk or whole milk. So that doesn't seem to make any difference. So what you can use as your spoiler, you can use vinegar. You can use this as all in the lab. You can use lemon juice. You are going to do it twice. And pick something really fun. I will tell you this, we used to, this is interesting, right? So the skim milk used to not work, and we used to be able to, with the Coca-Cola or Pepsi, we used to be able to curdle the milk. There are pictures online, they are all older pictures. We tried it this year and last year, and we could not get it to work. Any acidic fruit will work, so you can squeeze an orange into there. Oh, the oranges are so good. Actually, the cheese tastes really good when you squeeze the orange in there. Energy drinks work amazing. You do have to use more than of the orange juice, but it curdles it, and it's really gross. That would be what you would make a dice with, or cow, and then throw it away. Cranberry juice, apple juice, they both work really well. So you just have to think of a liquid that is acidic, and you just got to get the pH down to 4.5, and you got something in your house. Lemon, limes, and the eggs, I think I already talked about, you're going to denature the eggs in different ways and see which one works best and take a picture. And oh, I just thought I should have a slide about eggs and prostate cancer. So this is because I had just read the chapter from that book, How Not to Die. Highly recommend the book. It's amazing. I didn't read it from cover to cover. I like randomly pick a chapter. And so apparently this week, a year ago, was when I finally read the chapter on prostate cancer. I wasn't worried about for myself, but I had just thought I should finally read that chapter. So men who ate 2.5 eggs per week had double the cancer progression. So these were men who already had prostate cancer. They doubled how fast it metastasized. That is not a good thing. 81% increase, that means death. Almost doubled the amount of death. 100% is double the rate of death. From 2.5 eggs per week. Again, this is early stage prostate cancer. There was only one thing worse, and you can probably guess, because here's the reason the barbecued chicken was in the slide. Yeah, chicken, 4 times worse. Oh no, I'm sorry. Hey, I don't know, yeah, it quadrupled the cancer progression. Really? Oh, yeah, because when you have the combination, it has to do with the amino acids in chicken, and then the sugar and the high heat of barbecuing, that you get these guys made. Heterocyclic amines. There's a lot of them in poultry. And you're going, wait a minute. I gave up beef, and everybody says eat chicken. Oh, who tells you to eat chicken? And if you grill it, you're going to get cancer. And if you add sugar to the grilling stuff, which is what barbecue sauce is, you will get age. or you can eat. That's chard from my garden. That's my dog. I am not the short. I'm not like a little gnome. My dog is little. She's under 10 pounds. It's a cat. All right. Yeah, that's the chard that grows in my garden. You could just eat vegetables and then you don't have to worry about this. So your liver is trying to deal with this that's coming from chicken and eggs and meat, animal products. Plant products don't have this stuff. They don't have creatine. This is why my cat gets to eat meat, because the cat needs the creatine. All right. In case you forgot what age was, I'm assuming you all got this one right on the test. I hope it made you giggle and jiggle and wiggle and laugh. I was going to make a bonus for you to come up with your own acronym of what age stood for. Well, you can send me an email maybe and have fun and make it stand for something really cool. But this is like this crazy slide, and this is an even crazier slide. But the RAGE is the receptor for age. So whenever you have glucose, sugar, and protein, so sugar and protein, it's going to make this, and then it binds to the receptor, and you end up with oxidative stress and inflammation. And it pretty much cross-links to everything, your blood vessels, your organs. I don't know, what else is there? Oh, maybe your brain. And oh, just a reminder, fructose does this apparently 1,000 times better. That's a lot, 1,000. All right, you end up with, this was my favorite description of what age is: clumps of useless debris resistant to the body's digestive and cleansing processes. That doesn't sound good. Yes, basically it accelerates aging and diabetes. And there you go. This is different levels of age, advanced glycation end product in different products. And there's no comparison to roasted or barbecued chicken thighs with the skin. It was way out here. This is mama's milk, or an apple, or a tomato. These are like the baseline. You can't get rid of like these low numbers. This is a roasted yam. You should not have your oven temperature ever above 375, ever. You can roast things at a lower temperature. It still works, and you can roast without oils, which is probably a lot of the problem that happened there. Otherwise, this would be down here in like the 3 range. Make your own beans. All right, life cereal. Oh, but still, look what happens. Yeah, roasting. Oh, and cheeses start showing up. Oh, look at that. Cow's milk. Mama's milk. Cow's milk. That doesn't make any sense. Why people drink cow's milk. All right, because you were told to, right? It's not just what you eat, it's how you cook it. So endogenous means it's inside your body. So high glucose and fructose. So high fructose corn syrup, high levels fructose in your body. Exogenous is, oh, there you go. So cured meat. For every piece of bacon you eat, 9 minutes off your life. It's animal protein. So American diet, sugar and animal protein. Bacon, again, is a class 1 carcinogen. Just go and add plutonium instead. Would you really do that? No, of course you wouldn't. So why are you still eating bacon or salami? All right. It is how it's cooked also. Whenever you get to a high temperature, broiling, it's never been on in my oven, and frying, which is oil. Water cannot get up to a high enough temperature. You get 1,000-fold more. And also, unfortunately, barbecuing. So, experiment. They gave the same diet to 2 different groups. And the only thing that was different was how they prepared it. So the one group, they steamed it or boiled it for 10 minutes, and the other group, they broiled, I don't remember what it was, for 20 minutes. And the group that they gave them the broiled, whatever, 67% decrease in their arteries relaxing, and a similar increase in those age markers. That's not good. Let's keep it real, right? Animals know. Eat real food. And in case you've never seen this, again, anti-cancer. Look at that. They're beautiful colors. Eating Mother Nature and, oh, human-made food. So we talked about sugar. We talked about frying. And now I'm talking about animal food and how it is all prepared. Frying, high temperatures. Yeah. So again, you can do another challenge, another extra credit. And it's not so much the points for my class. It's the points for your life and for all the people you love. Just do it. Why not go for it? Try and be plant-based for 30 days. That's it, for 30 days. See what happens. All right. So what happens when you get age? Arthritis, kidney disease, atherosclerosis, 2.5-fold more prevalent in diabetics because of the high glucose. Neurological disorders, so you can pick dementia, 3-fold increase. Coordination loss, so Parkinson's. Neuropathy is a change in the sensation. And cancer, you can pick your type of cancer, pancreatic, breast, lung, colon, prostate. You can probably keep going on and on, every type of cancer. And we always talk about women and menstrual issues, but it's actually the men that have the greatest issues with this, probably because they eat more barbecue. All right, and your eye health. But we can fix that, right? Or we could all just start eating healthy and become like the person who enables our family. It's been really cool to see how many of you have gotten somebody in your family in on this too. So you don't have to preach. You can just show them the movie, right? All right, so eat food. It's even better than medicine. Don't eat this. This is prostate cancer, is the purple ribbon. This is not food. I'm supposed to have a red X that comes in and says, no more cured meat. Go completely plant-based for a month. You can do it. You all hopefully have watched the movie. And you're making a recipe, and I can't wait to hear what recipe. I'm not going to show you this video. All you have to do is Google TED Talks, and there is the reference to it. This was actually really cool. He talks about the problems of eating meat. He doesn't talk so much about all the health problems. Gotta see that in the Forks Over Knives. But then he mentioned something that was fascinating to me. Rowdy found it really rather disgusting. But you can actually grow meat from cells. And so rather than a chicken who's consuming the food that a human can consume and takes 6 weeks and causes all these issues, it takes him 6 days to grow chicken cells in a pot. So it was really, really quite fascinating. All right, so I will see you all soon. And Joey's trying to help me. Stop